The cell is the basic unit of structure and function in all living things. Without the cell, you wouldn't have life, because it is a reaction chamber in which all of the chemical reactions of life occur. Water is the solvent of life because it's a polar molecule. The DNA we talked about before comes in 4 parts. You have 2 of these are the nucleotides, adenine and guanine. They are purines. They have double rings with a phosphate backbone and a 5-carbon sugar called deoxyribose. They're all connected together and they make duplicate chains on the other side, duplicate copies of DNA. For every one of the purines, you will have a pyrimidine that will match up with it. The pyrimidines are single ring nucleotides, and the pyrimidines that make up DNA are thymine and cytosine. So the nucleotides of DNA are adenine, guanine, cytosine, and thymine. There is another nucleotide, and it's called uracil, and it's found only in RNA. DNA will make copies of RNA off of it in a process called transcription. And the RNA has matching with when the DNA opens up, it unzips, and the RNA that is made off of the copy of DNA will be a single strand, and it will contain uracil instead of thymine matching with adenine. Thymine always matches with adenine, and cytosine always matches with guanine in DNA. But when an RNA strand is made off of it, you have thymine that doesn't have anything. It makes an adenine off of it from one side. But on the other side, where it had the matching adenine, from it, the RNA that it makes will be a uracil instead of a thymine. So RNA is made of the same nucleotides, and it's copied off of DNA with the exception of uracil. RNA is single strand, it's not double strand. It doesn't have the long-term stability and ability to repair itself that happens with the DNA, because it doesn't have the matching strand to use as a foundation to copy off of. But RNA is used for the manufacturing of proteins. And as I said before, RNA comes in 3 types. The messenger RNA is copied by transcription from the DNA, and it is clipped into parts. The exons are put back together, the introns are left behind. The strand that has the exons put back together will have a 5' and a 3' end added, and then it will leave the nucleus to go out, and that's the messenger RNA. The ribosomal RNA and transfer RNA are also manufactured in the nucleus, and they are used to manufacture proteins. The ribosome has 2 subunits that will expose the codon that is copied to 3-unit piece, 3 bases on the RNA, 3 units called a codon. They are exposed to the anticodon, which is the matching bases on the RNA molecule. The RNA molecule brings in the amino acid. The amino acids are put together by enzymatic action at the ribosomes, manufacturing the proteins that are useful for the building blocks of the body. So the cells of all living things are basically just protein factories. They're small protein factories. And we are basically a colony of individual units that are basically protein factories. So all living things are made out of cells, and the cells had to be in isolation from the environment in order for the thing to carry out the chemical reactions of life. Inside eukaryotic cells, which is what we have been talking about because we've been talking about having a nucleus, inside eukaryotic cells, the packaging of these proteins happens in the changing and packaging happens in the endoplasmic reticula. They do modifications. The proteins get inside. As they move further away from the nucleus, this gets modified into a structure called the Golgi apparatus, and the Golgi apparatus will export out of the cell by a process called exocytosis, which requires energy. Any movement across the cell membrane that isn't diffusion of one sort or another requires energy, and that is called active transport. Water can get in and out of the cell by a process called osmosis. Osmosis is the diffusion of water across a semipermeable membrane. And osmosis lets water go from areas of high concentration to areas of low. If you have a lot of salt in the water, then that salt will take it up and it will make it have a low concentration. So the water goes from the other side of the membrane into the areas with high concentration of salt. So if you put your cells in salt water, they will shrivel up like raisins if you look at them under a microscope. If you put them in fresh water, they will swell and burst. Freshwater protists actually are able to survive by having what are called contractile vacuoles that pump the water back out regularly. And this is enough about cells for today. I will go on from here another time.